In this video, we're going to take a look at the internal structure of the muscle spindle and how that muscle spindle operates with respect to providing that afferent sensory information to the central nervous system. And we start here on the left-hand side where we see the overall structure of the muscle spindle with the capsule partially removed. And what we see here is that the muscle spindle has a mixed neural innervation. When I say mixed, is that it has afferent axons, and these are the axons when generating action potentials. These axons will transmit that information via those action potentials towards the spinal cord, but we also see that there are motor or efferent axons transmitting action potentials in the opposite direction towards the interior of this capsulated muscle spindle. And if we take a look here at the bottom, here we see the interior of the muscle spindle made up of these intrafusal muscle fibers. These are muscle cells like we have that makes up the vast majority of muscle tissue, but these are muscle fibers that are not contributing to force output of the whole muscle, but these are muscle fibers that do produce force. And much like the extrafusal muscle fibers, the gamma motor endings refer to the gamma motor neurons that transmit these long axons and will innervate the contractile elements located in the polar regions of these intrafusal fibers. And this picture only illustrates these efferent axons innervating one end, but we see that there are contractile proteins on the other polar end. It's just that this picture is not illustrating the efferent or the gamma motor endings innervating these fibers as well. So the intrafusal fibers with the contractile elements located at the polar ends, when these are triggered to contract because the gamma motor neurons have been activated, that will result in a shortening of the polar ends of the entire intrafusal fiber, and in doing so will actually lengthen the middle portion or the equatorial portion of the intrafusal muscle fiber. And when that equatorial region gets lengthened, the afferent sensory neurons will get mechanically disturbed, and when those sensors get mechanically disturbed, they will generate action potentials and signal to the central nervous system that there is something going on inside that muscle spindle. Now, there are three basic types of these intrafusal muscle fibers. The first type here is referred to as a nuclear bag fiber. And the bag fiber, there are two types of nuclear bag fibers, but the bag fibers are named because of the equatorial region that has a greater diameter than the polar regions, and it's this region where the nuclei of the muscle fibers are collected. So the dynamic nuclear bag fibers are the muscle fibers named according to the location of the nuclei, but are particularly responsive during a dynamic muscle activity, so during the actual stretch of that muscle fiber, whereas the static nuclear bag fibers tend to be active when the muscle is held at a new length. The third type of intrafusal fiber are what are called just simply as the nuclear chain fibers. You don't see that enlarged equatorial region because the nuclei are dispersed throughout the entirety of that muscle of that muscle cell, as opposed to being collected in the middle portion. So we'll start here with the 1A's, and the 1A's are the sensory neurons. So the red is the efferent motor neuron, but the sensory neurons, the 1A's, exist within all three muscle fiber types and are often termed the primary afferent fibers, and because they're wound up and situated along or around all three fiber types, whether it's static or dynamic, these ones are always active. The 1As, and being 1A, they are the largest and fastest transmitting sensory axons. The type 2s, which are slightly smaller and transmit slightly slower, are found wound up, as you can see here, around the nuclear chain and the static bag fibers. So you see that the dynamic fiber here is not innervated by the type 2s, and these are called secondary endings that are not active all the time as much as the 1As. In terms of the efferent connection, we see that the gamma motor neurons, particularly that are active during a static situation, so a muscle that's held at a new length, innervate the static bag fibers and the chain fibers, whereas the motor neurons that control the intrafusal fibers that are dynamic, the nuclear bags, only innervate those particular muscle fibers. So it's important to note here at the bottom in the red, we're talking about gamma motor neurons that help to regulate the length of these muscle fibers and hence maintains the sensitivity of these afferent axons. So in terms of understanding when these afferent fibers are active, we look at a summary of these results, these experimental results that focuses on 1A sensory receptor activation given a couple of different types of stimuli. So if we start at the very bottom here we see in this experiment a muscle is stretched, and so it's at this particular length, it undergoes a dynamic stretch, and then it's held at a new length, and that's what the shaded region indicates, is that it shows the period of time when that muscle is actually stretching. And so if we go up to the top of our figure and examine the activity when this is a passive stretch. So the top graph illustrates a passive stretch where there is no gamma motor neuron activity, so no efferent. So this is purely afferent, in that during the stretch we see that 1A receptors, 1A sensors undergo this activation and described as impulses per second. And so once that muscle achieves that new stretch level at the very bottom here and is held at that new stretch level, we see that the dynamic response drops off pretty sharply, but doesn't return back to the true initial level. So the change in length, this is a passive lengthening, it does generate 1A activation, and at the new length, 1A activation is still elevated, but not to the same level. So even though the impulse is transmitted drops off very sharply, it's still above, it's greater than the initial shorter muscle position. And again, remember, this is all passive stretching up here. Now on the next set of experiments, we still take the muscle through that period in which it's stretched, but that's overlaid with activation of the gamma motor neurons. And recall that when a gamma motor neuron is activated, the polar ends of the intrafusal fibers are activated. Those contractile elements will shorten and will lengthen the equatorial region. Remember, up here at the top, the equatorial region lengthened. Up here at the very top, the equatorial region lengthened, but only by way of the passive stretch. But what we have up here is that when we activate the static gamma fibers, we see that it still generates a similar dynamic response, but notice right before the actual stretch, when the gamma fibers were activated, we see that 1A activation elevates above the baseline. but the muscle was not passively stretched here. So without muscle passive stretching, the activation of the gamma motor neurons still increased 1A sensory activation because the equatorial region was lengthened. Now, when you actually passively stretch the muscle, then we see a very similar dynamic response that we had with the stretch alone. So the steady, and then once that muscle achieves a new length, the steady state response, the impulses generated along the 1As, is elevated because of that sustained gamma activation, static gamma activation. But when the dynamic gamma fibers, and only the dynamic gamma fibers, are activated, we see a very small increase in the impulses along the sensory 1As, not to the same extent as the static gamma fiber activation, but these unique motor neurons is just slightly above the baseline level. But but when that's overlaid on top of the actual stretch, the response along the 1As are considerably heightened. And then once the muscle is held at the new length, the steady state is still higher than the initial baseline, but because these are dynamic and not static, there's a gradual decay in the impulses. So the impulses generated along 1As still stays elevated above the initial baseline, but it gradually decays because these are the dynamic gamma fibers that were selectively activated. So in terms of the dynamic nuclear bag fibers, recall that with this bottom tracing, this is specific to 1A activation and not type 2, which are the secondary endings, which tend to be active more to a greater extent when a muscle is not actively stretching, when it's at its new length. So this ends our first video on the different types of intrafusal fibers.